we go. Record. Perfect. So, yes, hello to everyone watching the recording. The practice exam should have been posted. I recommend you try it out on your own before watching this. We're going to really talk about the answers. So, good luck. Anyways, thoughts on that? On a scale of 1 to 10, what would you rate the difficulty? Throw a number. Anyone? 7 out of 10 in difficulty? Okay. What do you think? 7 or 8? Okay. You? Okay. Okay. Yeah. How about you guys? What do you think about the difficulty? 7 or 8? Okay. Sounds good. Well, so I'll show you the answers now, and then you can calculate how many you got wrong or what the percentage was. So the letters beside the numbers to the right of the number, that's the correct answer. Take a couple minutes to do that, and then we'll get started on the questions. Okay, did everyone got their answers? Yeah. I mean, like on a thumb scale, how do you think you did? Okay, okay, like this, okay, okay. You? Okay, okay. And you too? Okay, sounds good. I'm just trying to get a sense of how everyone did here. So I guess we'll start. Here is question 1. So it's B. And how we're going to do this is it's going to be back and forth. I'm not going to stand here and lecture. I've done that plenty of times. Really want you guys to try to engage, interact with one another so that we can get the most out of this experience. Usually when I go through these questions, when I took this course, I would look at every option and say, okay, what part of this option is wrong? What should it be instead? That's how I studied, and that's how really how you can do better. So when it comes to option A, why is that one wrong? You can really just, you can blurt anything out and don't be shy, okay? And this won't pick up what you're saying. So you don't have to be worried about being embarrassed.
I could guess because I know there are like negative feedback loops. So I kind of imagined, okay, there's a lot of calcium. I assume it just, I don't know. That was just my thought.
That's a great idea. All right. So if there's a lot of calcium in the bloodstream, there are going to be signals to, oh, we don't want to take up more calcium. So I guess the renal section hasn't been discussed yet, but I'm sure Dr. White delved into absorption and the proximal convoluted tubule. But so the kidneys are known to filter. So the blood has calcium, and it gets filtered because it's a small ion. It will get filtered through the kidney, but there's a process called reabsorption regarding what gets filtered will actually be absorbed back. So there are some things that you do want to reabsorb back, such as glucose. Glucose will be filtered, but glucose is really important to the body. So it undergoes a lot of reabsorption. There are some things that you actually do want to excrete, so those won't be reabsorbed. But if you already have lots of calcium in your plasma, then there won't be as much of a need for it to be reabsorbed. That's why this one's wrong. So here we want to know what will not occur. So if you have high calcium, then you will have decreased reabsorption. So in order for this one to be not occur, it would actually be increased reabsorption. That would make it wrong, which would make it the correct answer. Does anyone know what bone resorption is? Yes. Yes. Mm-hmm. So yes, you've described what is essentially a common pitfall in understanding what resorption is versus reabsorption. So resorption, bone resorption, is there are cells called osteoclasts that will travel to the bone and they will essentially shed the calcium off of the bone. The bones are where calcium is stored. So these osteoclasts will go, they attach on and they start chewing away at it. And so you get these really small bone fragments made up of calcium, calcifications that are released into the bloodstream. So that would then increase plasma calcium. When it's bone reabsorption, not resorption, that's when high calcium would essentially be deposited and stored in the bones. So resorption would actually, it would be decreased. So that's why if you already have high plasma calcium, then it would be decreased bone resorption, not increased. So that's why this one is wrong and therefore the correct answer. Because here we're looking for the wrong answer. For C, yes, you have a question? So the proximal convoluted tubule is a part of the kidney. I believe this was in your slides. Maybe Dr. White did not go over it, but it's a part of the kidney that is involved in the reabsorption process. You'll go over this in the renal section, but just the idea is that if you have high calcium, you wouldn't want to be reabsorbing it. You know? And so if you're not reabsorbing it, then that's why E is also true and therefore not the correct answer. If you're not reabsorbing it and it's going through your kidneys, it's going to be in your urine. Okay. What about D? Why is this one wrong? Yes. Yes. If there is more calcium in the plasma, then what the body can do with it is that they can deposit it in the bones. That's reabsorption. Yeah. So resorption, or yeah, calcium deposition in the bones. Resorption, however, is actually breaking down the bones to increase plasma calcium. Yeah. So it's that's something that often catches people off guard. What about option D? What do you guys know about vitamin D? Yes? Exactly. So vitamin D, the active form, so it's 1,25 dihydroxyvitamin D3, that goes and acts on the small intestine and increases the absorption of calcium, dietary calcium. So if you have high plasma calcium concentrations, you don't want to be increasing your dietary intake of calcium. And so that means that you would want less vitamin D or less of the hormonal active form of vitamin D, because that one functions to increase calcium absorption from the gut. Does that make sense? I could explain it again. Yeah, okay. Okay. So to study for a question like this is a diagram that really kind of goes over everything. I recommend knowing this. Something I didn't include here is parathyroid hormone. So parathyroid hormone is a mediator of this, and it could very well show up on your exam. It's the general topic of that. Okay, any questions before we move on? No? Solid. Okay. So I guess until 7, we have a lot of questions left. So we'll try to go quicker. If you have any idea, just really blurt it out, okay? So why is option A wrong here? Don't be shy. Yes, exactly. Right? Which cells is it? Okay, that's okay. So it's insulin, it's what did you say? Exactly. Yep. So the beta cells that synthesize insulin. And so when you have type 1 diabetes, it's actually type 1 diabetes. Is it hyperresponsiveness or is it an autoimmune condition? It's autoimmune, right? So it's really T cell mediated. You don't need to know that, but it's the immune system through T cells, it's attacking the beta cells. And so then you have fewer beta cells, you have fewer insulin being made. That's why you see people who have to take insulin shots, okay? And what is type 2 diabetes? Is it autoimmune or is it something else? Exactly, right? So B and D are swapped. Type 2 diabetes really it could be caused by a lot of things, but maybe sometimes in the insulin signaling cascade, there's something wrong. So it causes a hyperresponsiveness to insulin. And how about E? Why is this one wrong? Exactly. So really, when you see absolutes and answers, kind of be skeptical. I'm not saying say it's wrong right away, but you should be skeptical of it. Does anyone know another hormone that works to increase blood glucose? Anything? It's okay. So epinephrine, also glucocorticoids such as cortisol. Okay, so just stuff to keep in mind. This is so the correct answer is C. And you can see here when diabetes goes untreated for quite some time, blood pH decreases. So you go into acidosis, that's what it's called. And you get more acetone and acetone gets converted into ketones. And so that causes ketosis. Okay. Question 3. Honestly, this one we can't even go over. So a lot of endocrinology is actually just memorizing what some different parts secrete. So I would be very familiar with the female and the male reproductive system and the intricacies of that. Okay, so really there isn't an explanation here. It's quite memorization based, and so you can refer to lecture 5 to get more information on that. Okay. So why is option B wrong here? Any idea why this one's wrong? You have any idea what this one's wrong? This was just on Monday. Almost. So the thing is, what happens at higher altitudes? Like think about the sun. Exactly, right? So say I'm in Vancouver or I'm in the Rocky Mountains, right? So I differ in altitude, and that difference can actually affect the amount of UVB light that reaches me, right? So if I'm at sea level, there's so much more atmosphere that could really take away from the UVB light that reaches my skin. Okay, but if I'm up in the mountains and the atmosphere is a bit thinner, then then that will affect how much UVB light gets to me and how much vitamin D can be produced. Okay, so it's not lower. Vitamin D production would actually be higher at higher altitudes, or be lower at lower altitudes. Okay. How about option C? Why is this one wrong? Yes?
Skin pigment, the molecule responsible for skin pigmentation is also responsible for the skin's like whatever goes in and out.
Exactly.
Device is also skin.
Yep. Yep. So melanin made by melanocytes, which influence how heavily pigmented your skin is, also can serve a protective effect against UVB light, which can actually influence how much vitamin D you're making. So rather than here saying that it's independent, it's actually dependent. Okay. And so I guess what we'll do here is that we'll move on to option E here. So African Americans and Caucasian populations, they differ in their skin pigmentation. And so where African Americans are more heavily pigmented, they have more melanocytes, and this actually prevents UVB light from synthesis of vitamin D and subsequently this hydroxylated form. Okay? And so that's apparent in the graphs in your lectures you had on Monday. You can see that. I'm sure Dr. White showed you the discrepancies between African Americans and Caucasian populations. And what about D here? Is it independent? No, it's dependent. It's dose dependent. So if you have more circulating 25 hydroxy vitamin D, there is more antimicrobial peptide production by macrophages. And then for option A here, you can refer to this graph. See that, so CYP27B1, that the mRNA, this encodes for a protein, and this is the 1-hydroxylase enzyme. And so that takes 25 hydroxy vitamin D, adds a OH group, so it's a hydroxylase, adds an OH group to 25 hydroxy vitamin D to make 125 hydroxy vitamin D. Yes. Yes.
He says a lot of other things that are not on the slides during the lecture. Should we just really, I guess, study from the slides and then refer to what he's saying?
So for example, like tell me as to what like he says, like that aren't on the slides. Okay. I felt that really often you can get the answers from the content in the slides. It never hurts. If he elaborates more and that helps you understand, then definitely refer to that. I do encourage watching lecture recordings to help you study. So, you know, lecture recordings versus just looking at the slides, the recordings are much better because Dr. White really does explain what you're seeing on the screen. Okay, so I would pay attention to what he says, but if it seems off topic, then it likely is. Or if it seems too nuanced, it likely is. So that's the end of endo, but you realize I've gone over a lot of things. I like oversight over a lot of things I haven't touched on. So you should be familiar with the thyroid hormone cascade and like how iodine is involved with that. You should be familiar with Cushing's disease and Addison's disease. You should be familiar with the zones of the adrenal glands that are making the mineralocorticoids and the glucocorticoids. Okay. What else is there? I mean, we didn't touch on the male reproductive system. Anything I'm missing? Feel free to jump in. What else have you learned that I didn't touch over? Let's see. Well, yeah. So we'll move on to the respiration section. So it's been a while, so I can see how these questions might be a bit difficult. But anyways, is option A true? Yeah. Does anyone know approximately how many times less the resistance is compared to the systemic circulation? Like a number? Yeah, it's 10 times less. Okay. Option B here, this is kind of, it can get this nuanced. This is kind of a memorization option here. You do need to know that serotonin can cause smooth muscle contraction, which will increase pulmonary vascular resistance, but then acetylcholine will do the opposite, vasodilation to decrease the resistance. There's a slide on that in the lectures. So why is option C here? Why is that one incorrect and therefore the right answer for this question?
My process was like ventilation. I made that it was like ventilation blood flow.
Mhm.
That blood flow is greatest at the lungs, and therefore a greater length denominator would make the overall ratio smaller. So
Yep, exactly that. You can, when you think of the lungs expanding, I know Dr. Lauzon, she made an analogy to a slinky, right? So if you hold a slinky up from the top, the furthest ring will be far, or the most upwards ring will be farther from the second one, but it gets, the distances between them get smaller and they get very compact towards the bottom. Okay, so that's essentially how the lung alveoli are expanding. Towards the top, they expand a lot more. So towards the top of the lung, there's more ventilation. But then you learned in cardio and in this respiration section that blood flow is quite heavily affected by gravity. And so there's more blood flow towards the bottom of the lung. So if we do a ratio of ventilation to perfusion, you know, the ventilation's the greatest at the top of the lung, where the perfusion is the highest. So that would be at the top of the lung. The ventilation perfusion ratio would be greatest at the top of the lung, and it would be lowest at the bottom of the lung. Okay. And so option E, this is straight from the slides. It's actually just the exact definition, but this is the graph you can refer to for the answer to that one. So you see here, the bottom of the lung, this is the ratio. It's lowest here, but as you get to the top of the lung, it's the highest. And the optimal gas exchange ratio is around Rib3, and where the ratio is 1. Okay. So this one, maybe someone can just give an explanation as to why it's C.
Not necessarily getting everybody.
Yep. That's you probably described it better than I could. Right. So when you're undergoing cellular respiration, everything you learned in general biology with the electron transport chain and how you have to consume oxygen, and then a byproduct is carbon dioxide. So increased metabolism will lead to more carbon dioxide being produced and less oxygen being available, or more oxygen being consumed. So that would decrease PO2 while simultaneously increasing PCO2. But then if you're not changing alveolar ventilation, there are two things happening. You're not intaking as much oxygen and you're not getting rid of as much carbon dioxide. So that's why you'll see that the partial pressure of oxygen is going to decrease, whereas the partial pressure of carbon dioxide is going to increase. And so I recommend you go over this table. I literally just chose this option, but you should be familiar with how all of these work. I recommend that. Okay. Okay. So, anyone know what this effect is called? What is the theme that this question is asking about? It's kind of like a physiological phenomenon. It's like a scientist's name. Yeah, it's the Bohr effect, right? So sometimes when it comes to studying for this exam, there are just things that you can't really fit in your head. Often I like to really draw. Like when it comes to like an hour before the exam, I'll try to draw as much as I know, like the graphs. And you just label the top of the graph. And when you like right when you start the exam, if you draw it, you know, okay, that's the Bohr effect. If I use it, great. If I don't, it's just there, but there's a chance I don't use it. But then if you get a question, you don't need to dig in your mind because you already drew it down. So the Bohr effect, this is really what happens. It's when you have increased temperature, partial pressure of carbon dioxide, and more acid blood pH, then the graph will shift to the right. However, if the conditions were the opposite, the graph can just as well shift to the left. So that's what this question was asking about. You won't need to know what DPG is for the exam. It was never asked and never will be, but it was just a part of this graph, and this graph shows it quite nicely. Okay, so here, increased temperature, increased carbon dioxide, and increased hydrogen ions, so increased acidity. Let's see here. If we decrease blood CO2, you would expect the shift to be left because the Bohr effect says that if it's increased, it'll go to the right. Okay, so this one's wrong. Increase in temperature will cause a shift to the left. No, we just saw that it's going to cause a shift to the right. And then C and D kind of contradict one another. So if you get lucky with that on the exam, you can cross these out. So indeed, it's none of the above. But I really do recommend knowing how to draw this and understanding that as well. Okay. Any questions? Take that as a no. Next up, this is also something you should be able to draw. So what do the neurons in the lower pons, what do they do? What's their function? If it's not to generate the basic respiratory rhythm, does anyone know what they might do? You have any idea? No? Okay. So the neurons in the lower pons, they actually work to promote inspiration. Okay. The cells in the upper pons, they work to turn off inspiration. And here, cells in the lower pons. Oh, I have these both here. That's interesting. Yeah, so this is to actually turn on inspiration or to promote inspiration. And so then I guess for these two, you can refer to this graph here. So you see that when, for option D, question 8, for option D, when you cut the vagus nerves and when you cut below the upper pons, it causes apneustic breathing. So this right here, this rhythm, this is apneustic breathing. And so you can see the vagi are cut and we've cut right below the upper pons. So this is where the lesion is and the vagi are cut and you get this rhythm. Okay. But then if you see cutting the pneumotaxic centers for option E, cutting the pneumotaxic centers in the upper pons causes breathing, I made a typo there, breathing to be fast and shallow. So if you cut the pneumotaxic centers in the upper pons, it actually causes here slower and deeper breathing. Okay. So I really recommend knowing this because this could easily be tested on. They could even ask you, like you see here, this rhythm is very similar to this one. And they might be like, oh, what lesions might cause the same respiratory rhythm? So you would say, okay, cutting above the upper pons and cutting the vagus is the same as cutting below the upper pons and leaving the vagus nerve intact. That's something they could definitely ask, okay? Any questions? Okay. Halfway there. So did you touch on emphysema and pulmonary fibrosis in the lectures? Yes? Yes? Okay. So what type of respiratory disease is pulmonary fibrosis? Is it obstructive or is it restrictive? Restrictive? Okay, very good. And what about emphysema? It's obstructive, right? So that's why option E is wrong. So when you think of pulmonary fibrosis, what do you think of? Like, what do you think of like the texture of the lung? Very hard, right? So like you could think of it as like sometimes elastic bands are like super loose, but every now and again you get that one that's like super rigid. And like no matter how hard you pull, like it seems like it's gonna break if you pull any harder. So that's pulmonary fibrosis. But if you get a super loose elastic band and you can like you stretch and you almost kind of feel like it's shredding apart and it's not going to return back, that it's very compliant, that's emphysema. Okay. So here, fibrotic lungs have higher compliance than normal lungs for any given pressure. Why is that wrong? They have lower compliance, right? They have lower compliance, but they have higher elastance. Normal lungs are more compliant than emphysematous lungs, but less compliant than fibrotic lungs. Why is this one wrong? It's kind of ranking them based on, okay, right? So you're saying it's switched? Yep. So normal lungs are actually less compliant than emphysematous lungs. I can't say this word never, but they're more compliant than fibrotic lungs, okay? Emphysematous lungs have greater elastance than normal lungs. Why is this one wrong? Yep, they have less elastance, okay? So when it comes to, you can really see this from this graph here. This graph shows on the y-axis, you have volume in terms of vital capacitance. And then on the x-axis, you have pressure. So if you remember what compliance is, you actually see compliance is it's the change in volume over the change in pressure. So, yes.
Can we understand elastance as similar meaning to resistance? Or like elastance is more like the capacity of coming back to its own.
Yeah. Yeah, I would say that. Yeah. Yep. Anyways, thank you. Good question. So when it comes to doing volume over pressure, if you have your y values as volume and your x values as pressure, then you're just finding the slope. So compliance in this case would actually just be the slope of these curves. And you see here that emphysema has the highest slope, meaning it's the most compliant, right? But if you're more compliant, you're less elastic because that's the relationship, because elastance and compliance are reciprocal. So it's really, this is high yield. They could ask you this, okay? So that is the end of respiration. Now we're on to cardio. Cardio is 10 lectures out of the 22. So you can expect around half of the exam to make up cardio. So you really need to know your cardio stuff, okay? So to answer this question, I'll ask you, how does the propagation of the signal start? Where does it start from? In the heart. SA node, right? Yep. And then where does it move? It moves through, yes, but before that, before the AV node, where does it has to move through a chamber of the heart. Which one is it? The right atrium, right? So then it reaches the AV node, and then it has to go through the bundle of His, and then the bundle branches, and then the Purkinje fibers, and then the whole thing contracts. Okay. So if you look at the ECG here, this is when the action potential in the right atrium starts. So this is right atrial depolarization. Where did it go? Okay, laser pointer. This is right atrial depolarization. And then because because the AV node and the bundle of His and the bundle branches and the Purkinje fibers, they're relatively small structures. So when you see a depolarization in them, these leads don't actually catch it all too well. So you don't really see it. It's happening, but you don't see it. But then once you start to activate the ventricular muscles, because these muscles are large and they take up an anatomically large, relatively large part of the body, compared to the AV node, bundle of His, bundle branches of Purkinje fibers. That's why you see this large deflection. But if you remember the activation sequence, you know that those have to be activated before the QRS complex, because the QRS complex is when the ventricles contract. So it would be between the P wave and the QRS complex, okay? Any questions on that? Okay, so they could really ask you what is being depolarized during the P wave, and they could give you 5 answer options. And you would say right atrium. They could ask you what's happening during the T wave, and you would have to know. Okay, so really have to understand this. Okay. We'll answer this question by asking you a question. Why is it the SA node? Yes.
Has the segment itself, so it has to build it up so big, whereas most of the, not most of, but let's say the ventricular muscle, it has to reach a specific threshold before it goes up.
Yep.
And if the circulation factor.
Yep. So all these other parts of the heart, they can quickly depolarize, but the SA node is making its own rhythm. All the other parts depend on proximal cells and the local circuit currents around them. So it really comes down to the biochemistry of it. Here you see that the SA node and the AV node, they have calcium ion channels, but they don't have sodium ion channels. And so sodium can really rush into the cell super quickly. If you think about the extracellular matrix, the extracellular fluid, it's mostly sodium. So there's a high electrochemical gradient. When you open up a sodium channel, it really loves to rush in. So that's why you can get this super quick depolarization that you see in these areas of the heart. However, because the SA node and AV node, they only have calcium, that's why it's quite slower. Okay. So this one involves the cardiac cycle diagram, or the Wiggers diagram. You need to know this thing like the back of your heart. Really, there's a million different ways to ask this question. So you really do need to know this. Now, why is this one incorrect? So why is this one the correct answer? Because it's false. Does anyone know why? It's the lub sound. So did they talk about lub dub and like that lub is the first heart sound and dub is the second one? Okay. Excellent. So that is the case. Here you see that it's the first heart sound corresponds with the mitral valve closing, and the first heart sound is lub, and here I have said dub. I'm not sure if you need to know if lub and dub are the first and second respectively, but you should know that the first heart sound corresponds with mitral valve closure, whereas the second one corresponds with aortic valve closure. Okay. Let's see here. Yeah, you can get very like nuanced and detailed with this. So you really should know the cardiac cycle, and I can help you with that. Any questions on the Wiggers diagram? Okay. No. Okay. So this is a bit of a memorization question as well when it comes to the first 3 options. The SA node does not have alpha adrenergic or nicotinic receptors. They're muscarinic, so that's what gives this one away. But why is D wrong? Yes? No, you got it. Okay. Anyone? No, no idea. Okay. So a beta agonist, an agonist essentially stimulates the beta adrenergic receptor. And so the beta adrenergic receptor works to increase blood pressure by increasing contractility. So when you increase contractility, the heart beats harder and you're able to pump more blood. Okay. So the stroke volume is larger. Okay. And what about atropine? Why is this one incorrect? No, no idea. Okay. So atropine is a muscarinic receptor blocker. So it binds to the muscarinic receptor and it doesn't let acetylcholine bind. And so when acetylcholine is the parasympathetic nervous system, parasympathetic works to decrease heart rate, decrease blood pressure. So by having atropine, you're blocking parasympathetic signaling, which lets sympathetic take over. So that's why you are having increased mean arterial pressure because you're increasing the heart rate, right? And so you can refer to this slide here that talks about it. Any questions? Okay. So for this one, why would a diuretic not work? What is diuresis? Did Dr. White talk about that? Yes, exactly. Right. So you can kind of see the word uretic or urine in here. So diuresis actually means to induce like peeing. So when you do that, you're diminishing, you're deplenishing your body of fluids, of water. And so then you're decreasing your plasma volume. And so plasma makes up a lot of the blood. And so if you have less plasma volume, you have less blood, you have less stroke volume. And that's why your blood pressure will actually decrease if you pee more. And so this is a very deadly low blood pressure. So you don't want to have it go any lower, And so antidiuretics, however, would be a correct answer because that prevents you from excreting fluids. So that will maintain your plasma volume up higher. You'll have more blood. You'll have more of a stroke volume. But diuresis, that's not the case. If you have a parasympathetic agonist is essentially you're stimulating an inhibitory signal. So that's why that would not work. And a beta adrenergic antagonist, you're inhibiting a stimulant. So it's kind of the opposite. We talked about this in the last question. Beta adrenergic would focus more on increasing the contractility of the heart, so to increase stroke volume. So you would want that to bring this blood pressure up, but here an antagonist would actually inhibit that and would probably kill your patient. Yep, so it's important to pay attention once you're doctors, if that's what you want to do. So here, alpha adrenergic receptors actually are on the vascular smooth muscle around blood vessels. And so what they will do is once they're stimulated, they'll cause contraction of the smooth muscle, which will increase the total peripheral resistance, right? And so here you can see that blood pressure is a function of heart rate, stroke volume, and total peripheral resistance. So if you can make the blood vessels shrink, you can increase the TPR, which will then increase the blood pressure. Okay. This one kind of self-explanatory. You do need to know what the renin-angiotensin-aldosterone system is. It's wonderful, and there's so many ways you can actually ask questions about it. So essentially, this is the slide you'll refer to. if I were you, I would be very comfortable drawing this diagram as well. You need to know the three arms and at the points of inhibition. So I mean, you have entire huge pharma companies that are making so much money off of these just by understanding how the system works. Okay, so should really be aware of the pharmacological inhibition of the RAA. Okay. Last question. So exercise, you should expect something on exercise. I can't guarantee it, but in past years it's been quite common. So essentially, again, you should be able to draw this diagram, should be able to really understand it. So you see that the heart rate goes from around 60 to 180, whereas the option here is that the heart rate becomes double that at resting state. No, it's triple. Okay. When it comes to stroke volume, it does increase a little bit, but then as you start getting up to the maximum 900, it wanes off a little bit. So here the stroke volume triples. No, that's not the case. Okay, it doesn't go up to 270. That's impossible because when you're exercising, your heart rate gets faster and faster and faster. So you don't have as much time to fill up the heart with blood enough to actually increase the stroke volume. So generally, when you start off, yes, your stroke volume is getting more, but you get to a point where heart is beating so fast that you can't fill it up in time until the next beat. So the cardiac output does actually triple, and so that's due to the tripling of the heart rate, not because of the cardiac output. The cardiac output is, sorry, the stroke volume. So it really is pretty much the same. Increases just a little bit. Here, when it says diastolic blood pressure greatly increases, you see that it does not really increase. It's more the systemic blood pressure that increases, which is bringing up the mean. And then the total peripheral resistance greatly increases. That's not the case. Here you can see that TPR actually decreases by about 60%. Okay, so when it comes down to the atriovenous oxygen difference, it does go from 5 to 15. So that one's true. That's why it's tripled. So you should really know how to do this. Okay. I guess that concludes everything. Thanks for coming. And I hope you enjoy your break and study hard. I hope this was a useful experience and you'll kind of know what you don't know, which is always a great place to start. But there's a lot of things I didn't go over. So for example, for cardio, you should know about, let's see, orthostasis. You should know about what orthostasis is. Other things include like the anatomy of the heart. There might be like a very basic memorization question about the different parts of the heart. When it comes to respiration, I know we talked about the Bohr effect, but you should also be able to talk about the Haldane effect, and also there's the thing where how open the blood vessels are at different parts of the lung, like the pressures. Yeah. Anyways, I think you know what I'm talking about. I can show you super briefly what you should focus on. Here, go here. I think it's this one. Oh, I would also know how to calculate different lung volumes. There might be a question on that. Like they'll give you values and you should know how to calculate that. This is very important to know. It's a very easy question to ask. Like in the pulmonary veins, what is PO2? What is PCO2? Or at different parts, okay? So you should know these values. Make flashcards if you must. Here, I don't think it's in this lecture. It's probably in this one. Yeah, Starling resistor concept. This could easily be asked, okay? Just gonna make sure I covered everything Dr. White might ask. So let's see. Yeah, you should know anterior pituitary versus posterior pituitary. Which ones, like anterior pituitary will make, you know, gonadotropin releasing hormone. Or that might be the hypothalamus. But anyways, it's the posterior pituitary that are more neurohormones, okay? You should know the classical, yes, know the classical definitions of these. This could easily be talked about. Let's see what else. Yeah, this stuff. ACTH, that could also be asked about. Make sure everything. Oxytocin, thyroid hormone, yeah. Pretty much kind of try to know everything to the detail that I provided in those questions. Yes.
Slide for, okay, he doesn't go into detail about, he doesn't really go into any of these. All he kind of, like, I think this kind of goes back to what, like, your question from earlier, but like, he doesn't necessarily say everything that's on the slides and he's throwing other stuff that's not on the slide. And so I don't know, so what he said about the thyroid hormones, so they increase metabolic rate and they help neural development. But he didn't mention any of the rest of this. So are we expected to just know that like two big ones, which he mentioned in class, or like all?
I think you should focus on what he said more. But when it comes to thyroid hormone, the big one is the effects on metabolism, the basic metabolic rate. Yes. Okay. Thanks for asking that question. Anything else? No? Okay. Well, you're free to go. Thanks for coming and best of luck to you. You got this, okay?